Okay, right. Hi. So predicting genes and functions in silico. In silico means on computer. That means you use a computer program to predict whether a particular sequence has a gene or not. So basically, these are some of the things you look for. You look for open reading frames. Open reading frames is what I mentioned earlier, a stretch of DNA that can be translated without encountering a stop codon. Now, um, if you don't believe me, you take an intron and then you try and translate it, and soon enough, after about 5 or 6, you will encounter a stop codon. But if a particular piece of DNA can be translated for some time, maybe 15, 20 amino acids, and still you can't find any stop codon, that is a good indication that what you have is actually a coding sequence. Exon prediction based on intron-exon boundaries, intron-exon signals, evolutionary conservation. You look across different organisms, and we have eyes, cats have eyes, dogs have eyes. So the function is, well, maybe they may have very specific functions individually, but in general, you have the genes that absorb light, you have the genes that control the ions, things like that. So you have conserved functions that can be comparable between different organisms. And then the next section talks about mm, the gene ontology project. I mentioned earlier what is gene ontology. Um, uh, Basically, it's saying that you're talking about information that is done in various different labs in across the world. So obviously, when they found something, they want to name it, and they gave different ways of doing it. Gene ontology helps this by giving a standard, so that a standard way of naming things, a standard way of annotating, so that people across the globe can actually understand each other better. Um, this is box 7.4 talks about what you do. Like I'm sure you've done blast searches before. So sequence homology, global alignment against local alignment, what that means. Have a read, have a go at it, and then see if you can understand it. Examples of sequence pairs, suited. What? There you go. Suited to global and local alignment and so on. Okay. Carrying out sequence assembly in complex genomes. Okay. Uh, if you talk about sequencing nowadays, right? Yeah, you can do sequencing, but in order to get higher confidence that you are getting the right sequence, you may want to sequence them again. So that means double, or if you want even more confidence, if you see that there are variations between different drugs, you sequence them again, sequence them. So the more sequence you have for a particular sequence, for a particular region, the more confident you are about the sequence. And this has been known as the sequence death. Not death, but death. So that means here, up here, so that means how, if you do a population study, and then you'll find that half of them is A and half of them is T, so you know that it's a polymorphic sequence. But if you see that in 3 individuals you find an A, but 200 other individuals will have a T, so there must be some sequencing errors and things like that. Scaffolding and contigs. Basically, you have small contigs. Small contigs are joined by scaffold, and then this provides the framework for the chromosome. These are just terms. Carrying out assembly in complexes, assembly statistics. We will not go into that. And this is the subject of bioinformatics. Why does it get stuck? Okay. And then we go into basic gene expression analysis. Now, I don't think we want to go. Okay, that will be the subject of of videos further down the road. Okay, so I'll see you later.